All right, so on to chapter 4, how cells obtain their energy. So again, try to keep in mind that we're building from the small to the large, from what we need a microscope to see to what is almost too big to see, entire ecosystems, the earth as a whole. So we've looked at individual atoms and molecules. Excuse me. So now we're going to look at how those molecules are actually incorporated into our bodies. So, all living things on the planet need energy to sustain themselves. We'll look at how plants obtain their energy, which is different than how we as animals obtain our energy. So we'll be looking at that in the next chapter. But this chapter, we're going to focus on basically how animals get their energy. Something like a hummingbird, being a small organism, excuse me, and having a really high metabolic rate means they use a lot of energy. Does anyone know, for example, how many times a human heart beats per minute? So on average around 60 to 70 beats per minute. Anyone know how many times a hummingbird's heart beats per minute? Give this a second to load. Listen to the ad. Da, 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 da. All right, so skip that. So we've, probably hopefully you've all seen hummingbirds, maybe not going in slow motion. How many times their wings beat per second? Their heart actually beats around 220 or more beats per minute. So easily 4 times the rate of humans. And so for their body size, because their metabolism is so rapid, they need a lot of energy. So pretty much all day while they're out and active, they're looking for food. The problem is that when night comes, most flowers close up at night, or a lot of them do anyway. And so there's a lot less food available at night. If a hummingbird were just to kind of sit on a perch overnight and sleep and wait until morning to go feeding again, it would end up dying. It would literally starve to death because of its body's demand for energy. So hummingbirds actually have to kind of go into a little type of hibernation every night. It's actually not hibernation, it's called torpor, Torpor. But it basically is like hibernation. Their bodies slow down, their metabolism slows down so that they can maintain themselves until morning when they then, start feeding again. So I think we get the idea here, right? So go back to the hummingbird here. So they're kind of unique in that they have to hibernate every evening, otherwise they would starve to death. But basically, what we're going to be looking at is bioenergetics, which is how energy flows through living things, like hummingbirds, like humans. So there is a branch of science called bioenergetics. And if that was something you were interested in, it would be looking at how energy flows through living things. We'll look at how nutrients cycle through living things. They're a cycle. A plant takes in phosphorus, for example. It uses it to make new roots, and then eventually the roots die. Phosphorus is released back to the soil. and then it's available for another plant. So nutrients like phosphorus, potassium, nitrogen are cycling, whereas energy goes one direction. So from the sun, ultimately through plants to make carbohydrates like glucose, through the animals, the food chains, on from a higher concentration to a lower concentration. And so we'll be looking at that. And so basically metabolism, which kind of biologically is a bunch of chemical reactions that ultimately take energy in and use it and convert it to a different type of energy, typically from a more concentrated type of energy to something like heat, we give off heat as our bodies exist, and heat is a less concentrated form of energy. And so that energy again flows from the sun, through plants, through animals, through decomposers that break down living things once they die, on ultimately out as heat. We're going to look at photosynthesis in a little more detail in a future chapter, but here's an important equation. One of the, maybe the only, but one of the very few equations that you are going to need to know for this class. So I'm not going to focus on it now because we're going to pick it up again later. But basically, you have CO2 and water that are taken in by plants, CO2 from the air, water from the soil, and then ATP, remember from previous chapter, that's adenosine triphosphate. It's the energy source at the cellular level. And plants use those those molecules and energy to make glucose, which is the C6H12O6. And they also give off oxygen, which is great for us because we breathe in oxygen. So we do have a very important relationship with plants, not only in that they provide us with food, the C6H12O6, but they also provide us with some of the oxygen that we need to breathe. And again, we'll be coming back to that equation several more times in the next couple of chapters. So there are two basic metabolic pathways within living things. There are anabolic pathways, where we take these individual small molecules represented by those green circles, and we build larger molecules that our bodies need for various purposes. And of course, it takes energy to do that. So our bodies take in these individual molecules through the food we eat, and then we use our body's energy to build larger, more complex molecules. Complex carbohydrates, for example. And those are anabolic pathways. We also have catabolic pathways in our body, where we take those large molecules and we break them down into small ones that our body can use in that way. And in the process of breaking the bonds that hold those individual elements together that make that large molecule, we give off energy. So it takes energy to build large molecules, and that's an anabolic pathway, and it releases energy or gives off energy in the catabolic pathways. So building is anabolic, breaking down is catabolic. And so the process that facilitates these pathways, these chemical reactions, are proteins called enzymes. And so in lab this week, if you're in the lab, we will be looking at enzymes and measuring some of the different things that affect how quickly an enzyme actually works. So in the process of studying energy and energy transfer, we call that thermodynamics. bioenergetics, how energy flows through living things, thermodynamics, study of energy and energy transfer, thermo meaning energy, and dynamics meaning change, movement. In other words, maybe transfer. Two laws of thermodynamics that we're going to look at. And the first one being that energy isn't created or destroyed. And sometimes this is a tough one for people to kind of get a handle on. There always has been, since the universe was created, the same amount of energy. We don't have more energy, we don't make it, and we don't have less energy, we don't destroy it. So that's what the first law of thermodynamics deals with, that energy isn't created or destroyed. The second law of thermodynamics deals with the fact that when energy is transferred from the sunlight into a leaf of a plant, or when that plant is eaten by an animal and that energy is converted into meat, or when that animal dies and is decomposed and broken down, that transfer of energy is really very inefficient. There's a very low rate of efficiency. Typically, it's only about 1%. If you start out with 100 units of sunlight, you only get about 1 unit of chemical energy from that sunlight. I'm going to take a quick pause here, and we'll get right back to some more aspects of energy. All right, back at it. I had to take a pause there so I could get something for my throat. I mentioned before I've got some issues that kind of mess with my vocal cords, but anyway, so we've been talking about energy but haven't really defined what energy is yet. And it's simply the ability to do work. So it takes energy to do work. And when we say work, we're not necessarily talking about like our whole body. These kids pedaling around on their bicycles is, it's doing work. It's work that the kids need to be able to actually pedal. So their bodies need energy to do that work. But we're also talking about the ability for our bodies, individual cells, so every cell in our body has to do work, has to build molecules like we talked about in a previous slide. And so that's work as well. So it's whether we're talking about work at the cellular level or the whole organism. That's basically what energy is. And I also mentioned heat, right? We talked about energy being transformed from one form to another, from more concentrated sunlight energy to the chemical energy inside of this leaf, the glucose, to ultimately kinetic energy, which is the doing work again. And along the way, a lot of the energy, as it gets transformed from a more to a less concentrated form, is ultimately given off as heat. Again, our bodies, animals in general, need, as a byproduct of their bodies doing work, the cells give off heat. And so basically heat is still a type of energy, but it's so dispersed that it really doesn't do work, and it's lost during the process of energy transforming from one form to another. And in the big picture, this whole process of energy transformation is referred to as entropy, which is basically a measure of the disorder in the system. And by that we mean, maybe you've heard chaos theory. And what that deals with in general, again, is that you're going from very concentrated, very organized energy in the sun, and then as that energy is transmitted across the cosmos, and a small little percentage of it actually lands on the earth, and that energy is used by a plant along with water and CO2 to make glucose. That energy becomes less and less organized, or in other words, more and more disorganized and more chaotic, less predictable. So that's kind of what we mean by entropy. So there's two basic types of energy: potential energy and kinetic energy. The water that's behind a dam, like Shasta Dam, for example, has a lot of potential energy. And we use that potential energy. In the case of Shasta Dam, for example, we use that potential energy to spin turbines that make electricity. So the water that's just sitting there behind the dam has potential energy. It has the potential to do work. It's not doing work yet, but it has the potential. Then when the gates are opened up and the water starts flowing through the tubes that ultimately spin the generator, then that water has kinetic energy. It's actually doing work. So still water, lots of potential energy. The water that's flowing through rivers and creeks has kinetic energy. Even this water, even though there's not a hydroelectric plant, this water is still doing work. It still has kinetic energy. Think about what this water is doing. One thing that it's definitely doing is every rock that this water flows over is being smoothed, slowly getting smoother and smoother because of the like sandpaper type action of the water just flowing over the rocks for decades, hundreds of years, thousands of years. And so over time, any rocks that are in a waterway become smoother and smoother because of that water's kinetic energy. So I guess I don't have my book in front of me, but this figure's in the book. And the question is, with each of these items, what type of energy or what type of process is happening here? Do these processes take energy, require energy, which would be endergonic? An endergonic process is one that needs energy for it to occur. And then there are exergonic or exergonic processes, those that actually give off energy in their effort to do work. Keep in mind, though, that even exergonic reactions still need a little bit of energy to start the process occurring, and that energy that is required to start the process is called the activation energy, the amount of energy to basically start breaking the chemical bonds that result in releasing that energy. So with these four items, we've got basically A up there, which is more or less like a compost bin. So is the process of composting requiring energy or releasing energy? Again, if you want to take a little more time and think about these before I show the answer, you can always just hit the pause button and think about it, and then go ahead and start it up again. But basically, composting is an exergonic process. It's actually giving off energy. The input, could be the sunlight that warms up the compost bin, that speeds up, starts speeding up the process of breaking down organic matter. And in the process of breaking down those bonds of those more complex molecules, you get the release of energy. If you compost, one of the obvious types of energy is heat. Composting, if you're doing it right, gives off heat. Sometimes even compost has been known to kind of spontaneously combust, although it's not truly spontaneous because we know it's the buildup of heat to the point where it's possible that it could actually reach the flash point. where it starts burning. I jumped ahead here, but B, the process of a chick hatching, is an endergonic process. So that chick, to be able to grow and develop inside of that egg, takes energy. So the parent, the mother, is the body of the mother is taking all that energy that it eats to build the embryo, to build the shell. It's feeding the embryo as it's developing until it gets to the point where it lays the egg, and then that chick continues to develop until it hatches. But that's an endergonic process. What about making a cup of tea? Letter C, cup of tea. It's an exergonic process. So again, it's giving off heat energy, for example. And then what about the silly thing there where you got these human hamster balls? It's also an exergonic process. So again, that's kind of like the water behind the dam. If you're just in that hamster ball at the top of the hill, there's potential energy there. And so then that little bit of activation energy to push that person off the edge of the little slope there, then that becomes kinetic energy. And so, if that ball, that human hamster ball, got to the bottom of the hill and, ran into a person, there would be a lot of energy there, a lot of kinetic energy. And if it bounced off another ball, it would be releasing energy. Or eventually it would stop, but it would still be releasing energy the whole way down. So figure 4.7 kind of shows you visually the relationship between energy and the pathway. So in the graph, I'm gonna hit the escape here. Again, I keep losing my cursor, and for the life of me, I still haven't figured out why I can't get it back when I have my slideshow running. But anyway, so again, we're looking at activation energy here. The y-axis is energy from a little bit to more. Notice there aren't units on here, so basically it just goes from none to more. And then the pathway over time. And if we look at the purple line, which would be the reaction if an enzyme weren't present. So remember, going back several slides, we were talking about enzymes being the molecules that basically facilitate biochemical reactions. We call that catalyzing. So if we look at the purple line, we've got the reactants, and so it would take that much activation energy for this chemical reaction to occur, whatever it is. And again, this could be any number of hundreds or thousands of chemical reactions that are happening in our body alone. And basically what we're looking at is comparing the difference between the two activation energies. And so with the enzyme present, whatever enzyme is required for a particular reaction, you have basically less activation energy that's required to get the same end product. And basically what that's doing then is it's saving our body energy. It's like getting having a sports car that gets 12 miles to a gallon, and then getting a really fuel efficient car that gets 40 miles to a gallon. Saving a lot of energy. And our bodies like to save energy as well. The less energy we're using, the more product we get in the end, the more efficient our bodies can be with what they're doing. So basically enzymes, definition, enzymes are molecules that catalyze biochemical reactions. And what that results in is they lower the activation energy. So they lower the amount of energy it takes for the reaction to occur, but it doesn't change the free energy of the reaction. So you still get the same end product. It just takes less energy to get there. So we all know that our bodies and all living things, especially warm-blooded animals, have a range of temperatures that our body needs to be at. If our body gets too hot, get sick and an infection, and you start running a fever, you may or may not know, but if a human's fever temperature gets to a point 103, 104, definitely 105, 106, you have to get that person's temperature down immediately. Otherwise, cells start dying, suffer brain damage, and then maybe get to a point where you just can't recover from that. And the same thing at the other end of the spectrum. People die from hypothermia. Bodies get too cold, that the cells in our body start freezing, and then they break, cells break, and you also can't recover from that. So most of the chemical reactions that are important to the cells in our body would happen way too slow at the temperatures that our bodies exist at. And so over evolutionary time, our bodies have, and other living things have evolved these enzymes that speed up the reactions. So again, these chemical reactions generally are going to be much quicker at higher temperatures. But our bodies can't survive at higher temperatures. So these enzymes allow the reaction to occur fast enough that it helps or that it works for us. But you don't need the higher temperatures to make that work. You've seen like food group pyramids or things like that kind of show the relationship of different food groups. Kind of looking at same idea here, but with enzymes, 5 food enzyme families. And so the figure shows kind of what those enzymes work on. So we've got an enzyme called cellulase, and you may know what cellulose is. Fiber, right? Fiber that's in plants, and we need a certain amount of fiber in our diet. Our bodies don't digest the fiber completely, but it kind of works like a cleaning tool inside of our bodies. And that's cellulose, the material that's in a plant cell. But again, notice this enzyme is called cellulase, A-S-E. There are a few exceptions, but if you see a word that ends in ASE in relation to, biology, it refers to an enzyme. Cellulase acts on breaking down cellulose. So there typically is some type of relationship there where you can kind of identify what that enzyme is actually working on. So basically the fiber from grains that we eat is the breakdown of that is facilitated with this enzyme. So again, it's a chemical, a process that helps speed up or facilitate the ability of our bodies to break down grains. Again, we don't digest them completely. We're not, our guts are different than, say, a cow, where their guts are designed to be 100%, plant-based diet. Our bodies don't have the complex gut designed to really fully break down plant material. But again, we still need that in our diet. Amylase is for helping to break down other carbohydrates. And we're looking at, kind of one pathway is breaking down vegetables, and then another pathway for amylase breaking down fruits. But still, carbohydrates, more simple carbohydrates, oftentimes fruit, and then more complex carbs in things like potatoes, starches, other veggies. We've also got another one that's not specifically on this list, but maltase is also for digesting carbohydrates, but in this case specifically sugars. So that enzyme focuses on that. We've got amylase. Lactase. Actually, lipase is in here too, a little narrow one there, and that's for breaking down lipids. So lipase, lipid. It's a little sneaky one there, but it's a little bottle of vegetable oil there. So that's for digesting fat or lipids. And then we've got lactase, which is for digesting dairy, which again, the chemical there, lactose. And we're actually going to talk a little bit about lactose and an aspect of it that you may be familiar with, the idea of being lactose intolerant, where your body doesn't produce the enzyme lactase. So for those of us who can't eat cheese or can't eat ice cream or drink milk, it's because your body isn't making lactase. So there are products where you can take the actual enzyme before you maybe eat a little bit of ice cream or something. And so you're artificially introducing that enzyme into your body so that you can maybe enjoy a little bit of ice cream or something. And then the other main one here is protease. So protease, protein. And so this is digesting the protein that we would generally eat in nuts and beans, and meat, of course. So these are just a handful of enzymes that are important in just the food that we eat. And you actually have an assignment that deals with this. So you've probably already worked on that particular assignment. Let me go back to the... So a little bit of anatomy, maybe, if you will, of enzymes. So first of all, enzymes are usually fairly complex molecules, and we'll actually see a real enzyme, but to make it easy just for understanding purposes, we typically show an enzyme like this. So the green kind of oval with a bite out of it would be an enzyme, like protease, amylase, whatever it might be. On that enzyme is going to be the active site. And then what that active site does is it allows a place for the substrate to bind to. So the substrate is the molecule that the enzyme acts on. So if this were the enzyme lactase, the substrate would be lactose. So we talked about some of the different examples there. So whatever the enzyme is, the substrate would be the molecule that the enzyme acts on. Again, every enzyme has an active site, and that's the location on or within the enzyme where the substrate actually binds or attaches to it. And so for the reaction to occur, the substrate needs to bind to the enzyme. Again, if we were talking about the substrate being the lactose that's in cheese, you eat cheese, it's got lactose in it. That lactose is looking for the appropriate enzyme. In this case, it would be lactase to bind to, so that it can be ultimately digested. If your body doesn't have that enzyme, then that substrate is still there and it's not digesting. It's not, your body isn't able to break it down because you don't have that enzyme. So the active site on the enzyme and how how quickly or how efficiently the enzyme does its job is affected by a variety of factors, and these are things we're going to be looking at in the enzyme lab that we're doing. But things like temperature, too high or too low of a temperature can certainly affect the ability of the enzyme to do its thing. If the pH in the cells are too high or too low, the concentration of salt, and some other things as well that can have an impact. The problem then is that when you have these extremes, whatever those extremes might be, let's say, for example, the temperature is too high, what can happen is that enzyme can actually denature. And again, we'll look at this process in our lab associated with enzymes. So think of, so here's first an actual sort of more realistic image of what an enzyme might look like. And again, it's a lot more complex than the nice little green, egg there that we use for a drawing. Whereas on that nice little diagram, the green egg with the bite out of it, you can see where the active site is because it's missing a piece, and that piece looks exactly like the substrate. On these more realistic images of enzymes, you can see by looking at it, there's no way you would be able to tell where the active site was unless you knew it. And it's really more of a chemical thing. It's not really even that it looks a certain way. It's more that it chemically is a certain way. So it's not even really like an empty space. Think of it like a lock and a key, right? The key, the substrate, is a certain shape so that it fits in the lock, the enzyme, to basically make the lock work. It's kind of the same idea here. So again, in a more realistic image of what an enzyme might look like, the actual active site isn't so obvious. But going back to the idea of denaturing, you've got a raw egg, and so that egg has different proteins in it, and if you decide to then apply heat to that egg, in other words, cook it, you heat it, and that heat causes the enzymes to denature. And in this case, it results in the egg becoming more solid. Once you've done that to the egg, you've denatured the enzymes, and there's no way to make it go back to the way it was. Once you've cooked it, it's cooked. You can't uncook an egg. And so that's what denaturing would do at the cellular level. The enzyme gets pushed past its happy zone, and basically then you change it in a way that doesn't allow it to do its job anymore. Maybe another thing or another way to look at it would be paper clips. You got these nice perfectly shaped paper clips. If you unravel them and twist them around and make little shapes out of them, you can probably try to get those paper clips back to look exactly like they did before. But once you've twisted and bent them, it's just about impossible to get them back to be this nice perfect shape again. So again, you did an assignment where you were asked to look at an enzyme and talk about what it does. Within our bodies, there are hundreds of enzymes that have important roles in keeping our bodies alive. But we've even then used enzymes and done some applied science to be able to use those enzymes to benefit us in other ways. Detergents that we use for washing clothes, dishes, things like that, enzymes have been harnessed to do that, to break down oil stains, break down certain types of stains that we may get in our clothes. Specific enzymes attack those stains and break them down. In textiles, we've used enzymes for getting all the different looks in clothing, like denim, polishing leather goods, making leather soft. That's facilitated by the use of enzymes. A lot in the food industry as well. Baking, brewing, things like beer, fruit juice, all kinds of things. Our enzymes are used to make products better, taste better, make things happen quicker, more efficiently. Even making paper, converting starches. Making paper involves cutting down trees, chipping up the wood, and then those small little wood chips, which are cellulose and starch, are broken down and then kind of reformed into paper. And enzymes actually help in that process to make it a lot more efficient, which saves money, makes a product that's less expensive. So that benefits us even economically. So we've got, again, enzymes, we've got the substrate, and then within that enzyme is the active site. There are a couple different models that we've come up with to explain how enzymes and active site and the substrates actually change or how that works. The whole process of the substrate binding to the active site, which then makes the enzyme, do its job. So there's what's called the induced fit model that basically explains how the enzymes and the substrate go through this process of transitioning to make the substrate more receptive for the substrate, to make the active site more receptive for the substrate. And then once that substrate fully, attaches within the active site, it's then referred to as the enzyme substrate complex. A couple of big words that just mean now the enzyme can do its job because the substrate is attached to it through the active site. So that's one model, the induced fit model. I mentioned that enzymes are important at every level of our cellular makeup for so many different important functions. And that's why it's important to have a good balanced diet, provide all the basic materials that our bodies need to do their job. But it also, unfortunately, explains a lot of times why people have problems, health problems develop because enzymes aren't being made anymore by our body. And if those enzymes aren't being produced, then things can't work the way they're supposed to. So the requirements at a cellular level from one cell to another vary. The cells in our liver versus our heart versus our kidneys versus our skin versus our blood, all of those cells have different needs. And those needs even change over time. So again, cells in our stomach versus the fat cells that put away energy for later, skin, blood cells, the digestive organs work harder following a meal than other times. So sometimes the enzymes have to be kind of kicked into gear. So there's no point in making something, for example, if you don't need it. So when you're between meals, for example, your digestive system isn't working, there's nothing for it to do. So there's no point in wasting energy making a bunch of enzymes to digest food you're not eating. But then when you start eating, or even when you start smelling food, the idea of your mouth watering, we've all experienced that. Smell something, or you're anticipating maybe dinner coming. That's because the response is that there's food coming. Your mouth starts salivating, which is producing amylase, which is one of the enzymes that starts the process of digestion, so just the liquid in your mouth, when you take a bite of food and you're chewing it, you're mixing it with that amylase, and that amylase is starting again that process of breaking that food down. So then that whole process, your brain working with your sense of smell to know there's food coming, then starts the process of getting your body kicking in and producing enzymes. So this process of regulating enzyme production, either starting it, promoting it, in other words, kicking enzyme production in because you're going to be eating, or after you've eaten and those enzymes are no longer needed, in other words, reducing enzyme activity, is done by a process called competitive inhibition. And so there's two ways this can happen, or two different models that might explain how our enzymes are regulated. Let me go back to this view for a moment. So in one model that's called allosteric inhibition, again, you have the enzyme, you have the active site, the place, in other words, where the substrate's going to bind to. Oops, hold my damn earphones out. Earbuds. So what happens here is somewhere on that enzyme is an area called an allosteric site. And so when that enzyme is going to be inhibited, when you, in other words, don't need it any longer, help if I put the right earbud in the right ear and the left one in the left ear, there we go. When that enzyme is no longer needed, say you've eaten already and you've started digesting your food, what happens is an inhibitor will be directed to the enzyme, and it'll look for that allosteric site, and the inhibitor basically will be shaped to fit in that site. And then the process of that inhibitor binding to the allosteric site causes the active site to change. And again, it's really changing chemically, but it's easier to understand it, I guess, if you think about it as a shape. So this inhibitor binding to the enzyme changes the active site so that this substrate can no longer fit in the active site, meaning the enzyme isn't going to keep doing its thing. So it's kind of stopped or blocked because you don't need it anymore. And so then time goes by, it's time for your next meal, your mouth-watering kicks in, and so your body's saying, okay, get ready, there's going to be some food coming. So what'll happen then is the opposite of an inhibitor. You get an activator that binds to that allosteric site, and then it goes and changes that active site back to be receptive to the substrate. So now the substrate can bind to the enzyme, and you get digestion or whatever the process is happening. So again, you get inhibition through an inhibitor molecule that changes the active site to stop the effect of the enzyme. Again, no sense of wasting that process if it isn't needed. Then when you need that enzyme again, you get an activator that will bind and change the active site back to make it suitable for that substrate. Again, some ways that we use this information is in, for example, the process of a lot of the drugs that have been developed. If you have high blood pressure and it's beyond what could be controlled with your diet, then your doctor might prescribe some type of a drug called a statin. And there are different statins, so this is kind of a category of drug. And so then statins are basically inhibitors of a certain enzyme, HMG-CoA reductase. And again, we know it's an enzyme because even though all that, it still ends in ASE reductase. And this is an enzyme that synthesizes cholesterol from lipids. So basically, this enzyme isn't maybe being produced in your body, or not enough of it's being produced to maintain your cholesterol at a healthy level. So by providing this enzyme through a statin, it basically is going to help reduce that cholesterol that's in your body. Tylenol is a category of drugs that basically is an inhibitor of a certain enzyme, cyclooxygenase, which basically relieves inflammation. It's an anti-inflammatory. And again, there's a lot of other drugs in that category. But what's interesting, I guess, is in this case, we didn't actually even originally develop Tylenol as an anti-inflammatory. It's kind of a side effect that we found out that it inhibits the enzyme, which results in inflammation. And so it does relieve it, but we don't even really understand exactly how it works. We just know it does, which ultimately that's all that matters, I guess, is that it does help. So there are lots of drugs, everyday common drugs that that are part of our normal medical treatment regime that are basically affecting enzyme production one way or another. So the process of inhibiting an enzyme when you don't need it anymore via making the site the wrong shape to fit the substrate, and then eventually activating it again so that the enzyme does its work. And sometimes there are intermediate substrates that ultimately get to an end product. So sometimes it's not even just one simple step. It may be multiple steps. But basically, this is a process of feedback, or more specifically, feedback inhibition, where the end product of the pathway inhibits something later in the process. And this is an important part of the way that our cells regulate themselves. For example, our bodies don't always produce ATP. Again, the basic energy at a cellular level. So when there's plenty of ATP in our bodies and we don't need any more, our bodies shut down the production of it. Again, it wouldn't make any sense to just make more of something than your body can actually use. It's just wasting energy. So then when our body starts detecting that we're running low on ATP, and that's kind of done indirectly because ADP concentration is higher compared to ATP concentration. When ATP, remember we talked about this briefly, when ATP is used to do something like move molecules around, in the process of that, one of the phosphate groups is broken off and that releases energy. That's how ATP molecule is used. Binds or goes where it needs to be. A phosphate molecule breaks off. So instead of being adenosine tri-3-phosphate, it then results in a molecule called adenosine di-2-phosphate. So when the ATP is being used, there's going to be more ADP the more that the ATP is being used up. So when our bodies detect there's more ADP compared to ATP, then that tells our body, okay, start making more ATP by catabolizing sugar, the basic, again, glucose or other sugars, carbohydrates that are in our body. So again, there has to be this feedback. Your body's producing stuff when it needs to, and then it stops producing when it doesn't need those things any longer. So let me go back to this. Get through that again. So here's some real world stuff. I believe I'm going to make this an assignment for this week. So here's kind of the setup. So I was talking about lactase a little bit ago, right? Lactase is an enzyme that's needed for specific chemical reactions in our body, specifically those that involve lactose, which is a type of sugar that's in milk and milk products, ice cream, cheese, etc. And again, you or somebody you know probably is lactose intolerant to some degree. And it could be, you know, minor. It could be just like, oh, I'm going to, you know, have a bowl of cereal with milk, and there may be just a little bit of something that causes, you know, upset, but not bad. Or it could be on the full other end of the spectrum where you get cramps, diarrhea, gas, bloating, just miserable immediately with just a bite of ice cream or a spoonful of ice cream. So again, those people that are lactose intolerant, it's because those folks' bodies don't make enough of the enzyme that digests milk. So these people are said to be lactase deficient, or again, we typically call it lactose intolerant. Although really, again, it's better to probably refer to it as being lactase deficient. Again, people can take supplements that contain lactase that helps them break down the lactose, and that can allow you to tolerate milk. There are all kinds of lactose intolerant products now, things that people can buy, dairy type, you know, milk replacement, ice cream, stuff like that, if people just got to have that stuff. So there are ways to get around that if you just can't live without one of those items. If we look at a map of the world, this shows with these different shades the percentage of people in these different parts of the world that are basically lactose intolerant or lactase deficient. So notice the trend that in areas that are these darker colors, so the southern third of South America, parts of Central America and maybe southern Mexico, southern part of Africa, Southeast Asia, and all, you know, Indonesia, all of these tropical islands out here in the South Pacific have the highest percentage of people that are lactase deficient. So with Australia kind of being an outlier, notice that, so imagine, you know, you got the equator through here, right? Kind of where I'm generally using my pointer. The farther north you get, the fewer people tend to be lactase deficient. So again, more tropical areas. So if you just look at, to say this, where it's at least half of the people. So again, from, you know, southern Mexico all the way through the rest of the New World, there are high percentages of people that can't drink dairy products. The whole continent of Africa is at least 40 up to 100% lactase deficient. Some of Europe, but mostly it's, you know, India, China, and then again, all of the tropical South Pacific islands have very high percentages of people that are lactase deficient. So let's see here. I think I lost my question. Let me take a quick peek here. I guess it pops up a little later. Sorry about that. So, so with that in mind, and again, you can always refer back to this image. The question that I'll post in the discussion board deals with deals with this question. Sorry, I'm getting distracted here by something going on out there. So the question is going to deal with basically why do you see this trend? Why is it that, people living in tropical areas tend to be lactase deficient at a much higher percentage than Europe, much of the northern, Russia, Mongolia, China, most all of North America? Why is this big discrepancy between these areas being deficient and these areas, being able to do dairy products? So again, this question will be in the discussion board, but this is some background that you can utilize to address that question. So let's get back to ATP a little bit. Again, our cells need ATP to do work, all the work that our cells do. But unlike, say, our car, where we've got a battery and that battery stores excess energy, the energy that our car may need to be able to turn the starter on, for example, but it's energy that's stored. And the battery is recharged by the car. Excess energy that's being generated by the car goes into the battery to make sure the battery's full. But our bodies, living things, can't store excess free energy. Now, we can store fat, and that can be broken down later. I'm not talking about that. I'm talking about the actual free energy that's made by ATP doing its job. Part of the reason that it can't store that energy is the process of storing ATP or any energy generates heat. And we just talked about why heat isn't good for our bodies. It can denature cells. It can cause the cells to be damaged to the point where they can't function any longer. So we basically have to make ATP as we need it and then shut down production when we don't need it. I mentioned in the previous slide that ATP is broken down by removing that one phosphate group, and then that release of the phosphate group where that phosphate group binds to another molecule to activate it is the byproduct of releasing that energy. For example, in our bodies, to make our muscles contract, whether it's our heart muscle or our leg muscles, whether you're a sprinter or a marathon runner, to make that muscle contract, you need energy. And so at a cellular level, it's that ATP, releasing a phosphate group, causing the molecule to activate, causing a muscle contraction. If we were going to get more into anatomy, we could talk about the difference between sprinters and marathon runners. Both depend on muscle contractions, but you can either have what's called fast twitch muscles like the sprinters have, or slow twitch muscles like the marathon runners have. You tend to be born kind of with more of one or the other. If you train as a sprinter, you can maximize those fast twitch muscles, but the guy on the left would never be as good a sprinter as the guy on the right, because the guy on the left clearly has a different category of muscles. He's got more of the slow twitch muscles. They don't need to be big and bulky. They just need to keep on twitching repeatedly for long periods of time, whereas the sprinter needs those fast twitch muscles to contract, but for a lot shorter period of time. So you can train and get better at one or the other, but it tends to be kind of just how you're born, more as one or the other if you were inclined to run at all. So we go back to look at the structure of ATP. Remember, adenosine triphosphate. You see the adenine group over here, a ribose group, and then the alpha, beta, and gamma phosphate groups. And remember, it's the release of this terminal phosphate group, the gamma phosphate group, that results in the release of energy. So when we end up with an ADP molecule, and then our body's rebuilding the ATP molecule, when you add phosphate back onto that molecule, it requires energy. So your body uses energy to build these phosphate groups and then to put them back on to make the complete ATP molecule. And then again, when this ATP molecule is set off to do work and it gives off the gamma or even the beta phosphate group, it releases energy called hydrolysis. So again, just chemically a little bit of what's happening there. It either takes energy to build these molecules, which we get in the food we eat, or when it's doing work, it releases that energy that can do whatever work we're asking it to do. So, two things that are a little more challenging in these upcoming chapters: the process of glycolysis, or what we call respiration, and then we'll look at the process of photosynthesis a little bit. So this is much more complicated than what we're going to talk about, because it is a very complicated process. All we want to do is get you to have a basic understanding of what's actually happening. And again, you can always look up, Google glycolysis and look at nifty animations that are out there. Again, sometimes when we see movement of molecules, it helps us visualize what's actually happening. And again, if you want to pause here, this might be a good place to take a break before you go on to the next section, but I'll keep plugging away. So this is one of those things that I think it would help you to draw it out. And you don't even necessarily need to draw it out in all of this detail, but some version of a diagram that helps you understand how this process flows. So we've got our glucose molecule, and again, this is simplified, right? C6H12O6. All we're focusing on right at this moment is the C6, the 6 carbon molecules. But remember, there's more to glucose than just 6 carbon molecules. Also, 12 hydrogen molecules and 6 oxygen molecules. But again, we're just focusing on what's going on with the carbon at this point. So again, this process is called glycolysis, which converts glucose into other molecules that our body uses. So the very first step is that we're breaking this glucose molecule into 2 molecules, ultimately 2 3-carbon molecules that are called pyruvates. Now again, to break a larger molecule into smaller ones, it takes energy. So for every glucose molecule, it takes 2 ATPs to start that process. And partly what we're going to be interested in as we follow or track this process is how much energy does it take and how much energy is produced. So first thing that has to happen is we need 2 ATPs to be produced somewhere to break this glucose molecule. So there's an intermediate step, and notice that remember triphosphate, right? And then we end up these 2 ATPs become ADPs because they've given off their phosphates, and where those phosphate go are to the 3 carbons. And then we're left with 2 ADPs because now the 2 phosphates from these ATPs are bound to this new molecule called glyceraldehyde 3-phosphate. Then it also takes some NAD and then ADP in the process, we're adding hydrogen ions. Remember, again, the glucose is going to have hydrogen. And so ultimately the bottom line is 2 ATP, 2 NADs, and then we generate a little bit of ATP as well. So we use 2, but we end up generating 4. So we get a net gain of 2 ATPs, going from a glucose molecule to 2 pyruvate molecules. So again, the net gain is that we get some ATP and we also get some NADH. All I really care about at this point is ATP is energy and NADH is basically energy as well, a different type of energy molecule. All right, so the first step, glucose takes energy to break the glucose molecule, and we end up with 2 3-carbon molecules called pyruvates, and we also gain 2 ATPs in that process. So now let's follow one of those pyruvates. So it takes first coenzyme A, so an enzyme, to work to further break down this pyruvate molecule. We generate more NADH in the process of breaking down this 3-carbon molecule. So there goes one of those carbons, and we're also then losing CO2, carbon and oxygen, from that original C6H12O6, the glucose molecule. And what we have now is a molecule called acetyl-CoA. So we've gone from pyruvate, which is a 3-carbon, have an enzyme going in here, and that's what this is. CoA is the coenzyme A. So that coenzyme A is attached now here. And then this leads us into the mitochondria, where the rest of the breakdown of that glucose molecule is going to occur. If oxygen's present, and we'll talk about if it's present or if it's not present, as long as we have oxygen available, we have what's called aerobic respiration. Aerobic, like aerobic exercise, you're using oxygen to burn energy. So if you have oxygen, you'll have aerobic respiration, which will allow that pyruvate to be converted to acetyl-CoA, and then it'll enter the citric acid cycle. So as long as oxygen is present, this process goes to what we call the citric acid cycle. The acetyl-CoA basically is the enzyme that is a carrier. It basically transports this 2-carbon molecule into the citric acid cycle, the CAC, citric acid cycle. So again, now we're in the mitochondria. Remember, that's one of the organelles, little power plant. So now you'll see why it's called the power plant of the cell. You have the acetyl-CoA, and again, I know there's lots of abbreviations here, and I'm never going to ask you to write out this whole overall chemical reaction, but again, we'll just look at the overview here. So you have acetyl-CoA, 2-carbon molecule, this CoA enzyme, and it goes into the Krebs cycle, which is the old word for it. Again, we call it citric acid cycle. Krebs and citric acid cycle are the same thing. Krebs is the person that named it, but then we renamed it. Of course, science always likes renaming things. So again, bottom line is what happens is that that molecule is broken down. It gives off NADH plus hydrogen ions, gives off another molecule called FADH2. So these are all the hydrogens that are in the glucose molecule, C6H12O6. So a bunch of hydrogens that are in that glucose molecule are are carried off in these other molecules. We also lose carbon dioxide at this point, 2 molecules of CO2. So that's given off as we exhale. It goes out through our cells, through our blood, and eventually out through our lungs. And again, this is carbon from the glucose, oxygen from the glucose molecule. And then it also gives off 1 ATP, so we gain another ATP of energy. So that's kind of the side where we're giving off molecules. Then as the cycle starts wrapping up, now that ADP that we generated earlier gets recombined with a phosphate molecule. We have 3 molecules of water. So again, this is water that's coming from the plant, taking up water through its roots from the soil. So we need water. And then in this case, it's, if it's us, it's in our body. So we're using molecules of water. And then we need an FAD and an NAD. And again, those cycle back around to pick up those hydrogen ions again when the cycle loops back around. So we're putting in that 2-carbon sugar with the enzyme, the NADH, FADH, or FAD are grabbing those hydrogen ions from the glucose molecule, giving off CO2, which too much of that can be toxic to a cell, so we have to get rid of the excess CO2. And then we're giving off an ADP. And then this side is kind of the recharge side. So these molecules are being brought in from elsewhere to replenish the cycle. So again, I'm not going to ask you to write out this chemical formula, but just have a general idea what's happening. So here's same thing that we were just talking about. We've got those NAD molecules, the FAD molecules, and we see them here basically through what's called the electron transport chain, which are a bunch of molecules that help the Krebs cycle occur. The NADHs are broken down. The hydrogen ion goes through the pump here and gives off these hydrogen ions. The FADH, same thing. The hydrogen ion is broken away and transported off. The ADP is added to a phosphate group to generate the ATP, and in that process we are picking up one of these hydrogen ions back to facilitate that process. So the smaller one here kind of shows the relation to the citric acid cycle. Again, you're giving off some of these molecules. The hydrogen ions are being broken off, transported through the electron transport chain, and then the ATP synthase is a complex that basically uses these hydrogen ions that are given off from the NADH, FADH to regenerate the ATP. So remember, the ATP is being used to do work. In the process of breaking the phosphate off, it releases energy. That energy is used to do something. Then those ADPs need to be sort of recharged, with a phosphate group. And so to do that, the ATP synthase complex grabs these hydrogen ions that were broken away earlier and then uses that energy to rebuild the ATP molecule. So we've got several things going on in the citric acid cycle, parts of it that are generating these hydrogen ions, part of it that grabs those hydrogen ions and resynthesizes ATP. And so it's the process of chemoosmosis that relies on the potential energy provided by that hydrogen ion gradient across the cell membrane to rebuild those ATP molecules. And again, I would really recommend just Googling electron transport chain, and you can see animations that show you exactly what's going on here. These animations are really helpful. As a matter of fact, maybe what I'll do is actually bring this one up. Give it a second to load there until my voice is going full frog here. So we're just about done with the lecture, but I'll take a little break and see which one of these electron transport chain videos pops up. This one's only about 2 minutes. So again, this kind of pulls the whole thing together. The glycolysis, which is the glucose molecule being broken down into the pyruvates. And again, you have the electrons being carried by the NADH that produce 2 ATPs. Remember, we saw that in the first section. The acetyl-CoA giving off CO2. Again, we got electron being carried by the NADH. That gets fed into the citric acid cycle. The NADH, FADH being the electron carriers, the hydrogen being the electrons. Again, we're giving off CO2, and ultimately, we got an emergency alert here. Evacuations up the highway from Reading. Fortunately, not this area where I am right now. So that whole process also gives off 2 ATP, which again, ATP we need to make energy, and this is how the ATP is being produced. And then most of it, though, is all of those hydrogens that are being given off from the C6H12O6 are being used in oxidative phosphorylation to ultimately generate more ATP. So again, big picture, you eat something, you have the basic glucose molecules that are being broken down in our body. And the reason that molecule or those molecules are being broken down ultimately is to make ATP, which is then going to do all the other work that our bodies need. Contracting muscles, digesting food, all of the things that our bodies need for them to be functional. The more we know about what's actually going on in mitochondria, the more we're able to, again, apply it using applied science. There are diseases of the mitochondria, and so now there's even very, very specialized areas of medicine called mitochondrial medicine. Doctors can specialize in treating diseases of the mitochondria. When your individual mitochondria aren't functioning correctly and basically you're not producing ATP like you should, you can have muscle weakness, lack of coordination, you can have things that appear to be like a stroke. It can affect your vision and your hearing. Again, I mean, this is like the power in your house goes out. Your fridge doesn't work, your lights don't work, your house doesn't function like it needs to without energy. If your mitochondria, for whatever reason, lack of enzyme, whatever it might be, aren't producing ATP like it should, then your whole body is going to suffer from that. So there is areas of medicine that focus specifically on those types of diseases. Okay, not sure why that happened. Now remember, that was all contingent upon there being oxygen present. Remember I mentioned in the beginning of that, the pyruvate molecule being carried by the acetyl-CoA. And as long as there's oxygen present, it goes into the Krebs cycle. If it's got oxygen, if it's aerobic, then the final electron receptor is an oxygen molecule. But there are conditions where the cells go through what's called anaerobic respiration. If there isn't oxygen present, or not enough oxygen present, then that NADH has to be reoxidized. And ultimately, bottom line is what happens is what's called anaerobic respiration. Instead of the process happening like it would otherwise, the process then is going to use a different molecule as the electron receptor. And ultimately what's happening here then is a process called fermentation. You may know examples of fermentation. Maybe foods you eat, for example, are fermented. If you like sauerkraut, for example, that's a food that basically has gone under fermentation. You put something with the right spices and so forth in like a sealed jar. So there's obviously not going to be enough oxygen in that sealed jar for regular cellular respiration to occur. So in that case, it's actually going to go through fermentation. In our bodies, going back to the idea of runners, maybe long distance runners, for example, although it can even happen in sprinters, the muscles are being used, right? The muscles are contracting for you to run or sprint. And it's possible and it's frequent that your muscles can become exhausted of oxygen. They've burned up all the available oxygen. And what happens then is you have this process of lactic acid fermenting in your muscles, which causes, heaviness and pain and so forth. So you get this lactic acid buildup, it's called. And then once that lactic acid builds up to a certain point, you can't continue using your muscles because they're not working the right way. So again, we call that anaerobic, without oxygen. And so respiration still happens. It's just a different process. Again, the process called fermentation in this case. So again, once we kind of started understanding the process of fermentation, and even quite by accident, we can go back about 1.5 million years if you look at the timeline on the left, where it says lactic acid fermentation is invented. I guess you could probably say it was more like it was discovered, because it was always there. It's just that we didn't know about it. So going back about 1.5 million years, humans discovered the process of fermentation. And whether it's animal materials, you see some different food items around the world there. Animal-based foods, plant-based foods, pickles, sauerkraut, some food that you may be familiar with, Vegemite from Australia, tequila, other alcoholic beverages, of course. But we basically, what all these foods have in common, just think about it for a second. Why, or what do all of these foods have in common, or how does the process of using lactic acid fermentation to make these foods, how did humans use it? Again, you can pause if you want to think about it for a minute. But basically, it's a way to preserve food. We can harvest cabbage, or we can harvest cucumbers, and we can eat them fresh. But 1 million years ago, we didn't really have a way to refrigerate stuff, right? We didn't have a way to keep food for an extended period of time. If you lived in a far north climate, at least part of the year when it's really cold, you could store food, just leave it hanging outside, and it would stay, viable for a long period of time. But what about, Africa and the Philippines and South America? Not much of a winter in those climates. So by discovering a way to preserve food, plant or animal-based food, we were able to have food at times of the year when it wouldn't normally be available, because we could ferment it, preserve it, and then eat it anytime during the year. Well, of course, probably the most important use of this is development of alcohol. I say important, tongue-in-cheek, right? We certainly could live without alcohol, but would it really be worth living? So, using this same process to ferment things in a way that not only did you maybe preserve it, but in the case of alcohol, you create something that has a whole different effect, right? I mean, we know what the effect of alcohol is. So it's not only maybe a way to preserve a beverage so that it lasts longer and doesn't go bad like water might. If water is just sitting around in a barrel, it can start getting algae in it and just kind of get to where it's not drinkable. But you make beer, wine, some other beverage like rum. Those are beverages that can last longer than just water. Especially important if you're out on a ship exploring the world. You need drinkable liquid. Water would only last so long, and you couldn't always find fresh water on places that you might find along the way. But you can make some sort of fermented beverage and take that with you, and basically survival. It's not so much about getting drunk as it is, this is a liquid that helps you survive when you don't have fresh water to drink. So this process, the process that results in alcohol fermentation, and we're actually going to do a lab related to this as well. You're going to, in lab, you're going to have a chance to make a fermented beverage, a root beer or ginger beer. And not only did we use this process to basically generate alcohol. So those pyruvates are broken down into carbon dioxide and the acetaldehyde. The acetaldehyde in anaerobic conditions, again without oxygen, uses the energy of the NADHs, the energy being the hydrogen ion, breaking off, ultimately then creating ethanol or alcohol. But so you got a beverage that has alcohol in it, so you get the warm fuzzy feeling of drinking the beverage and getting the buzz from the ethanol. But the other part of this is the CO2. Again, if that CO2 is trapped in this vessel that doesn't have oxygen, the CO2 can't leave, right? If oxygen can't come in, the CO2 can't go out. And so then it basically creates carbonation. Today, we just kind of inject CO2 into our carbonated beverages. But the first we discovered was actually using a natural process to make CO2 that made the beverage bubbly. So we made a carbonated beverage that also had alcohol in it. Again, in the absence of oxygen. If oxygen is present, then those pyruvates break down ultimately and generate ATP. But if oxygen isn't present, then we go through fermentation. And specifically in alcohol fermentation, we generate CO2 as we would anyway, but the CO2 is trapped. Again, if oxygen can't go in, CO2 can't go out. So we trap that in there and make a carbonated beverage that also is alcoholic. I think for the sake of time, I won't show this, but you can YouTube search for The Science of Beer. I'll just have it pop up so you can see which one it is. So it talks about the process of making beer. It's only about 6 minutes long, but again, we've been going for a while here, so I'd really suggest just going to YouTube and searching for The Science of Beer. Again, he just goes through, maybe most of you have been to Sierra Nevada or some other brewery, or maybe you make your own home brew. So maybe you understand how the process actually happens. But if not, otherwise, just go ahead and watch that little video clip there. So again, whether it's beer, whether it's fermenting grape juice, so we use grains and hops to ferment to make beer, right? We use grape juice and we ferment grape juice to make wine. The CO2 is the byproduct again. We don't want carbonated wine, carbonated champagne, but in wine, the carbon dioxide is actually bled off. There are release valves, and so the carbon dioxide can be, removed from those vessels because, again, wine isn't carbonated. The CO2 is produced anyway, but it's just bled off instead of being kept in there to make the beverage fizzy. And so we use, around the world, we ferment different things to make different products. Rum is basically using like sugar cane, or at least originally it was sugar cane. We ferment corn to make whiskey, those kinds of beverages. We ferment potatoes, or at least that's how it typically is, to make vodka. So you can start with different products, but it's ultimately the same process that you use to generate the end product. So we focused on glycolysis, but just kind of to wrap up this chapter, there are other pathways for breaking down other types of carbohydrates. Carbohydrates, some amino acids, glycerol, all go through a glycolysis, whereas fatty acids and some other amino acids go through pyruvate oxidation. Other amino acids actually go directly into the citric acid cycle. But ultimately that results in the end product, right? The oxidative phosphorylation that results in making ATP basically. And that's kind of the bottom line. Remember again, all of these molecules, whether it's carbohydrates, amino acids, lipids, whatever, are broken down in different ways through glycolysis, pyruvate oxidation, through the citric acid cycle, ultimately to generate ATP. And then that ATP is used to do work, make your muscles twitch, make your heart beat, all the stuff that happens in our bodies. I'm going to save this question. I'm not sure if we're going to do it as an assignment or not. So this is not an assignment right now. If an assignment pops up, it will be, in the discussions where all the other assignments are going to be. But it's kind of interesting to think about how people could have first discovered fermentation. Remember I said it was invented, right? The image said it was invented. But really it's more about it being discovered. The process was happening before we started harnessing it for our own greater good. To get drunk, basically, right? And again, to preserve liquids long term so you could go out exploring and have liquids so you didn't die from dehydration. But think about how it could have been discovered the first time. And again, this isn't an assignment right now, just something to think about. I may or may not make this an assignment, but if I do, it will be in the discussion board. All right, so that wraps up Chapter 4. We'll be on to Chapter 5 next time.